recording. So let's move on. You know you have Khan Academy if you want to improve your grade. Other than that, the Jupiter Schoology grade is the grade you have. When we're talking about, we've talked about, let's go ahead and quickly review, make an outline of what we've done already. You've done biomolecules. You've done cells. And you need to know this. You're gonna need to know this for your next test and for your final at the end of the year. All of it. All right. Cells. You've talked about how we make biomolecules. By the way, how are they made? How are they used? What atoms make them up? In cells, you talked about the different organelles. What they're used for, how they're used, where are they found. You talked about the different types of cells. Yes, bless you. And of course, you talked about what biomolecules are found in each organelle or made, which organelle makes which biomolecule. You've talked about different processes like photosynthesis. And you talked about how photosynthesis is autotrophic. In other words, they make their own sugars. How photosynthesis uses light. You've talked about atoms, elements, the chemical reactions. You've talked about concentration. You've talked about how things move across the membrane. And that means passive versus active transport. The different kinds of transport. You've talked about, let's see, where else? Where are we going with this? Kinetic energy, kinetic theory. You've talked about, in photosynthesis, pigments. What are they made of? And if you don't remember, proteins. You talked about separating molecules. Talked about homeostasis. Feedback mechanisms. Both positive and negative feedback. You've talked about cellular respiration. You've talked about photosynthesis using CO2, specifically carbon specifically carbon and CO2 that you've talked about photosynthesis producing oxygen gas and using carbon dioxide gas in the air. And it also uses water. You've talked about that CO2 is used as the source of carbon to make food, specifically sugar. You've talked about how water is the source, not sauce, source of electrons for the electron transport chain that's in photosynthesis. You've talked about that it uses light as a source of energy. Which reminds me, we've talked about kinetic theory of energy and the types of energy and what energy is, types, and what it is. Talked about temperature and how it impacts as an average kinetic energy. We talked about, where are we? In photosynthesis, we've talked about not just autotrophic light and pigments, but that you have two different reactions. They have photosynthesis happens in bacteria and in chloroplasts. And that in chloroplasts, especially, but you have, in any case, in chloroplasts, you have the light independent and the light dependent reactions. We've talked about that the light independent is also known as the Calvin cycle. And that the Calvin cycle is where you make sugar. The light dependent reactions are where you capture light, capture energy. And you put that energy into the electrons. So you capture energy. The light dependent reactions capture the energy and put the energy into electrons by way of electron carriers. That's right. And the carriers are in this NADPH, which is the final electron acceptor here. The light independent reactions also take the electrons from the light from the NADPH and ATP from the light reactions, the light dependent reactions. to make sugar. And of course, the light independent reactions that Calvin's cycle uses or captures CO2 by way of rubisco. Right? The most important enzyme, right? And we talked about let's see, where were we? When we're talking about biomolecules, I can't fit it in here. I guess I can zoom in. The types of biomolecules. I'm going to have to just start another one here. Proteins. Carbohydrates. Lipids. Nucleic acids. Where can I put this now? I have to extend it. Oh, that's not a good extension. Let's extend it with an orange line this way. With biomolecules, we've also talked about the 5 different kinds of biomolecules. We've talked about proteins, lipids, carbohydrates, I should say 4, not 5. I was thinking DNA and RNA, but obviously they're nucleic acids. That's correct. We talked about lipids and nucleic acids and carbohydrates. We talked about these things being polymers. That's right, except for lipids, which are not. We know that carbohydrates can have polysaccharides and monosaccharides. We talked about a bunch of different ones, but a few of the notable ones are obviously with monosaccharides. Let me separate these lines. With monosaccharides, the notable ones, obviously there's two really important ones. They're all very important for various reasons, but obviously glucose, I hope it's obvious that that's a big deal. And the other one is ribose. And I hope you know why, but if you don't, I will try to touch on it here. And then of course there's deoxyribose. With polysaccharides, we know we have glycogen, and that's A. And then we know we have B. That's one of the important polysaccharides we talk about. Cellulose is the other one. But there's a bunch of different polysaccharides. Just important to know that those are important ones. And why is glucose important for energy? Right, and it's in sugar. It's in our food source. It's food, right? And it's also makes, by the way, some of these are linked, so it also makes cellulose, right? And glycogen. It's involved in making cellulose and glycogen. Ribose, deoxyribose is found in DNA and RNA. So those are important. So, and I shouldn't have put that. Yeah, proteins are polymers. That's correct. They're also proteins make enzymes. They are the functional unit of life. They do a lot of work. They do a lot of the work, and they form a lot of the structure. Yeah.
What was that chain that was like a DNA thing, and you said the skeleton or something?
Phospholipid, the sugar phosphate backbone. Yeah, we'll talk about that in just a second. I'm going to expand DNA and RNA in a second. Enzymes, we talked about what impacts their function. That's right. We've talked about how we read graphs that look at their function. We've talked about something called denaturing, which happens usually because of the three main factors that impact enzymes, which is pH, extreme pH changes, temperature, right? And we've also talked, when you talk about function, though, we've also talked about something called concentration. Oh, I forgot one other thing, salt concentration. But concentration of enzyme and concentration of solute. Not solute, but it is a solute, but it's, oh my gosh. Ligands or substrate. All right, so it's a lot, right? We're not done yet. That's not all the stuff we talked about. DNA and RNA is, we know that we talked about their structure. We talked about their monomer. The monomer we know is either deoxy or ribose sugar. We know that there's a phosphate. We know that there's a nucleic acid. We know, not nucleic acid, nitrogenous base. We know that the nitrogenous base can be in DNA, can be A, T, G, and C. And in RNA, it can be A, U, G, and C. T gets changed for U. We know that in the structure of, there's a difference in structure of DNA and RNA. In DNA and RNA, you have, and there's commonalities in both. So maybe it's best I do a Venn diagram for structure of DNA and RNA. They both have a phosphate sugar backbone. That means they have phosphate sugar all the way down. DNA, however, forms a double helix because because it has two strands of DNA, hydrogen bonded together. Is that thunder? Huh?
Yes.
So one thing, there's like three things in the world I hate or afraid of. I'm not really afraid.
It's raining hard.
Thunder is I'm afraid, but not clowns. Clowns and thunder. I can't take it.
You said 3.
Did I say 3?
It's raining.
Clouds and, well,
Stay informed.
I fear God, but other than that,
Get informed.
and police officers, I guess, with weapons that are upset. But the thunder, well, not thunder, lightning, and clowns are the two things. I can't. I will slap a clown. Just let you know. Keep away from me. Oh, and I guess there's a bunch of stuff because dolls. I can't take dolls. China dolls are the worst.
I have China.
How did? Yeah, I knew that. I knew that you'd have a creepy little China doll. You probably have a whole room full of them on
No,
shelves.
I have stuffed animals.
Ugh. DNA and RNA.
I stuffed animals. Don't do that.
RNA has ribose, which has an oxygen. DNA has deoxyribose, which doesn't have an oxygen. RNA has a uracil, a U instead of a T.
Oh yeah.
So RNA is single-stranded instead of double-stranded. But they still have sugar phosphate backbones. They still have the same monomer. So that's ribose and deoxyribose. That's a short. We're going to get into that in this week. Tomorrow when we do transcription translation, we're going to DNA replication. Then we'll get into genetics. So that's the direction we're going. Lipids are long chains of carbon and hydrogen. They can be saturated or unsaturated. They can form things like steroids, which are carbon hydrogen bonds that are rings. These are nonpolar. All of these are nonpolar, which reminds me of something we should have added over here somewhere in the biomolecule section. Polar versus nonpolar bonds. We talked about all right. So now let's see. So you agree this is a lot, right? And you did all this in one semester. I'm very proud of you guys. You need to know that that's a lot for kids coming into ninth grade, and then you're hit with all this. Proteins do form polymers. They have monomers. We talked about the monomer of proteins. I think it's relatively important to remember that they have a central carbon. They have the carboxyl group, one of those functional groups that we talked about. They have a nitrogen, the amine group. They have a hydrogen, and then they have that R group, which is a variable, right? And that there's 20 different possible variables on that R group. That really is important when we start talking about transcription translation, and we start talking about it's really going to become clear why that 20 amino acids are important, because you have 20 options, and what makes a difference between what you inherited from mom and dad versus what I inherited from my mom and my dad is that code in DNA and the protein that it finally makes in translation. So there's a reason why it's important you understand that there's 20 different variables here. And we know that that they can be polar or non-polar. They can be ionic. They can be large or small. I'm just thinking about I know I'm leaving a bunch of stuff off, but I think I've hit all the highlights. Do you agree?
I agree.
All right.
In my opinion, it's a lot.
Yeah. Well, it's not a. It's just the beginning. Life is not simple.
No.
It's not simple. Storks don't deliver babies.
That's true.
You know, no, they don't. Santa Claus doesn't exist.
I never believed in
Yes, he does.
Life is complicated. There's rarely a simple answer when you're looking at how things work. And if you're gonna try to be a doctor to try to fix people or try to help people that are in trouble where the system starts to fail, you have to understand the basic way the system works and the materials of which the system's trying to use.
Mm-hmm.
And to understand diabetes or to understand how you see things and to understand, you have to understand even just something as simple as seeing, right? To see me, you have to understand light and how pigments interact with light and how you get electrons in the pigment shifting, which then send a signal through your optic nerve to your brain, which then allows your brain to process that signal, which interestingly ends up being inverted because we see things upside down. I don't know if you know that.
I do.
But when you look into, when your eye, the image that you're looking at gets projected onto the back of your eye, the place that's called the retina, the top rays of light are at the bottom, hit the bottom of the retina. The bottom rays of light hit the top. So all images are upside down. So your brain has to take a look at that, get those signals, and then turn it right side up.
Oh.
So your brain does all of that. Isn't that crazy? All these little signals. So a photon of light hits your retina, sends excites an electron, sends a signal, then your brain does all the thinking necessary to take that image and make sense of it.
Yes.
So what your eyes, the fact that we see anything and understand anything about the world that's around us, it's stunningly amazing, stunningly complex. Yeah.
Like, what if you don't have as good vision as other people? Like, how
That has to do, most of the time, that has to do with the lens. So just like any camera, this is kind of an off topic, but I think it's just good to understand so that you can see the complexity of what we're talking about here. So you have an eyeball, and at the back of the eyeball is a series of pigments, you've heard of them, called cones and rods, right? But they're just different cell types. These are made of cells, like everything else in your body. Here you have an opening in the eye. There's little strands of protective pigments. This pupil can dilate, open or close, just like a camera, letting more light in or less light. At night, the pupil dilates. In the daytime, it shrinks up. You don't want too much light in there because you get blinded, right? So this is the pupil, which suggests that your eye has...
Well, then use Google.
That looks wrong. That looks right to you?
Yeah,
Okay, all right. Whatever.
Serious.
I'm good.
How do you spell pupil?
Pupil. And she's trying to correct me. She gets me. All right, so anyways, the pupil lets in here, there's all this liquid. And so the pupil opens or closes. And by the way, when you're on drugs, I should suggest, and I don't mean illicit drugs. I'm talking about any kind of drugs that you might take from your doctor. It could be illicit drugs as well, but that's not the point. Here there's muscles. So that suggests if the thing is opening and closing, that suggests there's muscles. Well, you have tiny little muscles that pull and contract and relax. Little tiny muscles in your eyeball.
They help you close your eyes.
Of course, you also have muscles that help move your eye, right? So you're thinking the eye is pretty simple. It's just a lens, right? It's not, right? So any light beam that comes in from the bottom, any photon of light is going to hit the top. Any light beam that comes in the top is going to hit the bottom. So again, the image is going to be upside down. So if I have an image of an arrow, what's going to be projected on the back of the retina is a downward arrow. Because the light that's coming from this is going to end up the light beam is going to get hit here and then reflect downward, and then this light's going to go upward. And so what you're going to see is an upside down image. If that's all you did, but you don't do all that. You do more than that. There is a neurological disease where people have trouble inverting that. No, that's a different story. So this is there's an optical nerve that's connected. It's one of the big nerves. Your eye is an extension of your brain. So it's literally connected directly to your brain through this optical nerve. Has a bunch of these little neurons that come out and they go up into your brain, and your brain processes those nerve signals. This opening, right? There's a lens over it that helps you focus it like a magnifying glass, right? You need to magnify these things. You can't just so you have this layer of tissue. Let me do it kind of like this. You have this layer of tissue that sits out here, helps magnify the image so you can see clearer. Well, if this shape is perfectly concave, in other words, it's not going to be perfectly concave, then the image is going to be focused right here, where it's supposed to be. If it sticks out too much, which I think what happens to me, then the image gets focused here.
It's blurry.
And you see, and things are blurry to you. If it's a little flatter, then things get focused out here, behind the retina. Again, images are blurry to you. So what we do is we add these things called glasses. We add another lens that helps focus that image better. Helps make sure that the image shows up here on the retina and not here on the retina and not here in this space or behind. You want the image focused. When we're talking about images, we're talking about light. Light. We want light to be focused here. And this light to be focused not here, but right here. If you're nearsighted, it means you can see close up, but not far away. That would be me. You're as well. Then your image is either focused here or here. I can't remember which. If you're farsighted, it's the opposite. So you have to change these lenses. Corrective lenses have to be fixed. Now there is eye surgery where eye surgeons take this lens off. They take the lens off of you. They reshape it. They heat it up and reshape it to make it more concave or less concave, depending on what it is. The problem is, and they make it to the right shape so that it fits the eye better, and the image gets placed on the retina again.
Aren't you awake when that happens?
It is. They give you numbing agent, so it's not like they do it. There's pain involved, or like. Yeah, sure. said we're going to.
We did?
Yeah.
Okay.
We said, there has a few.
And then we know it's working, and these are nice tests.
Yeah.
Okay, we'll do that. And then 311, which you already have, 305, you already have. And then I just switched that to gray. And I think that's it.
That's it, yeah.
That's it. Okay.
Oh, well, assuming they organize it.
They're gonna take care of the people thing, yes.
I wanted to make it, I wanted to put it, I was gonna put all this in the handout.
I'm sorry, guys, I'm so sorry.
But consider that for a second.
How do you know what's that?
I'm so excited.
What if I'm medium sized?
You didn't pause it yet.
I can't, no, I can't see
You
far.
didn't pause
This
it.
time's up. It is for me.
That's
It
when
is.
you lie.
No, it's not.
You could, you're either near side or close enough where you can't see things. You want to talk to like that? Huh? Hold on.
I have this right
You
here.
said, I don't know what are you getting.
I got,
This
I got,
girl.
I got. Actually, I got this much nice on my
I got this right here.
That's probably the reason why you like this going to work. Yeah. My promise you, she gonna work just like the way you. I was like, oh my god. She always look at me like, yo, I was like, girl. Hey.
That's not her. You said you gonna help us.
I kinda wanted to put it together. Oh, I understand.
So hopefully it will be.
My thing is, I understand, I'm not upset with anything. Why are you all upset with me?
That's how I felt too, because then they're coming to me and I'm like,
Why are you, I don't care. Hey, look, if it flops, life goes on. But why are you upset with me?
Yeah.
You're the one getting all red and.
yeah.
All right, guys, sorry.
Wow, these looking is so good. You say I can.
Yeah, I know, I keep saying that. Woosah, woosah.
I can speak.
All right.